Dear friends and colleagues, if you are watching this video, it means that there was some issue with my flight, for which I deliver this recording, but I will be with you in a few hours. So I look forward for eventual discussion and comments. And if you want, of course, you can reach out to me asking the organizer my email address and also phone numbers if you want. This talk is about a very interesting, I think, exploration we have done recently, thanks to the academic teamwork also with the University of Ferrara, to which I belong, but also to the Harvard University and Stanford University, for which really it has been a pleasure for me to co-work with Professor Roxon and Professor Raffetto and others in exploring the origin of life, I would say, in a certain way. As per the picture, there is an exploration of the turtle on the surface of the oceans, where indeed life started. And what I mean by this is that if you watch this Britannica video, you are going to notice how the surface of the corals, where actually life began, is very similar to the surface of our endothelium lining. There are senior structures in both of these conditions that are sensitive to the different kind of flow acting on it, transforming the mechanical force of the flow into a biochemical message based on the movement of the senior structure because of the in and out of the different ions. If you look at this Britannica video again, on the surface of the corals, you will see a halo structure that is very similar to the halo structure I will show you at the end of this presentation for the first time up to our knowledge on the surface of the endothelium of the lymphatics. So on the surface of the endothelium lining, we already know in veins and arteries, there is a ciliar-like structure called Glycocalyx that does basically four things. And to remember what it does, I usually think about the PC, the personal computer. So permeability, P, protection, P, C, coagulation, balance, and C, contractility. So we have permeability regulation, we have protection of the endothelium lining, we have contractility regulation of the vessel, and we have balance on the coagulation. It's a very complex glycosaminoglycan and protoglycans structures that has been perfectly described by Zhang as a critical signaling platform that is involved, as you can see, in the migration, adhesion, proliferation, apoptosis, and many other biological characteristics. To make it simple, it's like a forest that is covering the endothelium lining, protecting it, as per this nice paper by Governor that is talking indeed about the vascular protective properties of the Glycocalyx. We know that losing the forest on the surface of planet Earth is not good for the Earth, as we know that losing the Glycocalyx on the surface of the veins and arteries is not good because it is associated with inflammatory conditions like thrombosis, chronic venous disease, and on the other side, atherosclerosis. Indeed, the Glycocalyx has been defined as a critical signaling platform, but also as an innovative treatment target as per this publication, for which we got very interested in it. And in honor of Egypt, I would say whenever we are getting interested in veins, we should also get interested in lymphatics. We know that they go together, like indeed Antonio and Cleopatra, like the inseparable couples, as has been defined also as a family affair in this very important paper by the beloved Hugo Parsh and Professor B. Billy. So we started wondering if there is such a thing as the Glycocalyx in the veins and the arteries. What about the lymphatics? Is there such a thing? And we asked all our friends around the world, and nobody has ever heard about that, for which we started our own investigation with this joint activity among different universities. It has been quite a challenge because harvesting the lymphatics, to look at them at the electron microscopy, is not easy at all. For this reason, I would like to congratulate the University of Ferrara Biolab, who has been able to use the sample we provided from ex vivo. So we are talking about the lower limb of human beings ex vivo, sent to the lab and fixed for electron microscopy analysis. So congratulations to Professor Raimondi for having identifying a proper fixation way that allowed us today to present to you what, up to our knowledge, is the first in a human electron microscopy picture of the Glycocalyx that, as you can see, is a halo structure really similar to the one we can find on the surface of the colons. And we are really happy to announce that now we are not impressed anymore because we published, so it's already published the paper on Scientific Report of Nature of this identification, together with, again, Professor Affetto, Professor Roxon, and many other friends and colleagues that co-worked together to provide these images at 100,000 magnification of the vein and of the lymphatic Glycocalyx, so that we can also look at them in comparison, looking also at the different microscopic features in terms of measurements that are related also to the different actions they might have in the veins and in the lymphatics, for which we can get involved in the discussion. But you could surely say to me that, Sergio, you're just showing us a picture. What are we really looking at? How can we know that it is the Glycocalyx indeed? And that's why we did immunohistochemistry, running some podoplanning staining to make sure that we have the lymphatics that you can see over here and here the vein, and also glypican staining that is confirming with the glypican that is indeed a component of the Glycocalyx, the presence of Glycocalyx-like structures. We also identified other components like brevican, mucin-2, and agrin. Now, brevican makes sense. It is part of the intersynapses tissue. Mucin is usually present in the extracellular matrix, so we're talking about the exchange of fluids. Agrin, we still don't know why it's there, and that's the beauty of the research. It keeps on going. And it's a research that is moving from the bedside, say in this case, back to the bench and then back to the bedside, because we are talking about edema formation. For sure, something that is not related just to the Starling principle, as per the teaching we received at the med school, but we know that indeed the lymph is different from the plasma composition, for which a sieve of various porosity, like indeed the Glycocalyx, might be involved in the creation of the edema formation. So in the lymphedema also, that is the hidden epidemic, and the phlebolymphedema that is putting together veins and lymphatics. Clearly a vital topic, I would say, that will be included among the many vital topics that will be discussed in Umbrella Island next May, where everybody is invited to submit vital topics for discussion. So feel free to reach out and to present your data, your ideas, your vision about what is vital we know and what is vital we develop as research lines for proper vein and lymphatic advancement. Thank you, and looking forward for being together in person.